All right, welcome back everyone. Here we are at our second lecture of Bio 139, our second lecture of the endocrine system, and today we will actually dive in and start our study of some of the glands and hormones of the endocrine system. Now, as always, we will begin with our not really attendance questions. What's the difference of an endocrine organ and an exocrine organ? So just kind of explain the basic differences between the two. What is glycogenesis? And when you get hot, you begin to sweat to cool down, or when you get cold, you shiver to warm up. This keeps your body temperature at roughly 98.6 degrees Fahrenheit. This is what type of feedback? Go ahead and try to answer those before listening to the correct responses. Hopefully you've done that. So let's see what the correct answers are. What's the difference between an endocrine and an exocrine organ? Well, endocrine, which is really what we're studying in this chapter, is a ductless gland. It does not have a duct, and it secretes its product, hormones, directly into the bloodstream. Exocrine organs, these are glands that secrete their product into some sort of duct, a tube, and then that tube or duct then spills out either onto the surface of the body like a sweat gland or into a hollow body opening like along the digestive tract. Next, what is glycogenesis? Now this goes back to Bio 137, so this might have been a little bit tricky, but glycogen, first let's talk about glycogen. Glycogen is how our body stores excess glucose. When we have more glucose than we need, we don't just urinate or poop it out. We keep it in case we might need it later, and we store this a few different places in the body, mostly in the liver. And the way that we store that excess glucose is sticking lots of pieces together and forming this long chain called glycogen. Well, that act of sticking glucose to glucose to glucose to glucose to make glycogen is called glycogenesis. And third, when you get hot, you start to sweat. When you're cold, you start to shiver. What is that type of feedback? That is a negative feedback loop. And that is most of the feedback loops in the body are negative feedback. And we will be talking about those extensively this semester, but especially in this chapter. Really, all of this is going to be important in today's lecture. So, without any more wait, let's go ahead and get to it. Today we're talking about the pancreas. And the pancreas is kind of weird in that it is an endocrine gland and an exocrine gland. It is both. It's going to serve both functions. And so what we see here in this image, that's the pancreas right there. It sits kind of below the liver, below and behind the stomach. It's a little bit hidden. It's shaped kind of, I guess, almost like a comma. It's a little bit curved. It's larger on one end, and it kind of tapers to a point going towards your left side. Now, the pancreas, if we were to open it up, it's got some ducts running through it. Pancreatic ducts are what they're called. And those ducts actually connect to the first part of the small intestines right as it leaves the stomach. This part of the intestines is called the duodenum. We will learn about that later this semester. So the pancreas is connected to the digestive tract at the duodenum by way of the pancreatic duct. Now, what we see over here on the right, This is a microscopic image of a part of the pancreas. And most of it is dark. Some of it has these little light staining areas. I'm going to go to another image that shows the difference in the dark and the light a little bit clearer. That's what we have right here. Again, here's the pancreas. We can more make out that larger area called the head of the pancreas, and that long pointy area, the tail of the pancreas. Here we can see the pancreatic ducts spilling into the duodenum. Now, up here at the top right, this is what I was talking about. We can see those dark staining areas and light staining areas. And this is where we get into the difference in endocrine and exocrine with the pancreas. Most of the pancreas, something around like 80 or 90% of the pancreas, is this darker staining area. These are called acini cells. Acini cells, and they are the exocrine part of the pancreas. So the way acini cells work is they will produce different digestive enzymes. The pancreas is actually responsible for a lot of the digestion. It produces enzymes that break down things like fat and protein and starch and nucleic acids like DNA and RNA in the food that we eat. And also they produce something called bicarbonate. And bicarbonate is a buffer. Remember, a buffer is something that acts to kind of prevent too much change in pH. When something is very acidic, a buffer kind of acts like a base. When something is very basic or alkaline, a buffer kind of acts like an acid. So when we pour that bicarbonate here into the small intestines, right as the broken down food is leaving the stomach, well, that stuff, that it's called chyme. The stuff leaving the stomach is called chyme, and it's pretty acidic. It's got all that stomach acid in it. So we need to kind of tone it down a little bit. And that bicarbonate, when it mixes with the chyme, it makes it not quite so acidic anymore. So all of that, the bicarbonate and those various digestive enzymes, come from the pancreas. Specifically, they're coming from the acini cells. And since they are emptying into a duct and into a hollow body opening, that's the exocrine part of the pancreas. The acini cells are the exocrine part. And again, that's the majority of it. That's like 80 or 90% of the pancreas. But scattered here and there are these lighter staining areas, and these are called the islets of Langerhans, or the pancreatic islets. Islets of Langerhans, or pancreatic islets. And this is the endocrine part of the pancreas. Now, if we just look at this under a microscope, these cells look pretty much the same. And when you see them in the lab, they're going to look pretty much the same. But in reality, there's a few different types of cells there. There's alpha cells and beta cells and delta cells. You do not need to be able to visually distinguish between alpha, beta, and delta cells. But you do need to know, what do they do? And again, this is the endocrine part of the pancreas. It does not have anything to do with digestion, and it does not empty into the pancreatic duct or the duodenum. This empties into the bloodstream directly. Remember, endocrine. So let's go through and talk about what do these endocrine portions do. All right, so when I am going through here, we are going to fill out the outline together. Now, in endocrine, I've pretty much filled out your outline for you. That will not be the case throughout the semester. I have filled out your outline pretty much for you, though. So first thing we're going to talk about is what hormones does the pancreas produce. We're going to talk about the stimuli, the function. In some cases, we're going to talk about disorders. So you do need to know those, and even though I filled out your outline for you, mostly you are going to have to add a little bit of material. So we said that the islet of Langerhans, this is the endocrine part. There were alpha cells, beta cells, and delta cells. We're going to begin with a hormone that most people are familiar with, and that is why we're covering things a little bit out of order than they are presented in your textbook. I wanted to first begin with something that we're at least a little bit familiar with, and that's insulin. Insulin comes from the beta cells in the islets of Langerhans. The beta cells produce insulin. And insulin is an amino acid based hormone. So remember, it will only be able to target cells that have receptors for it because it's an amino acid based hormone. What is it that causes it to be released? Well, first, a rise in blood glucose. When your blood sugar goes up, amino acid hormone insulin is released. So when your blood sugar goes up, insulin is released. But that's not the only thing. Increased amino acids, increased fatty acids, or acetylcholine. Think about what acetylcholine was. That was the neurotransmitter used by your parasympathetic nervous system, the PNS. So PNS activity rises in blood glucose, rises in amino acids or fatty acids in the blood, will all stimulate amino acid based hormone insulin. Why is that important? Well, those are all the things that happen when you eat. When you eat and you're digesting, your parasympathetic nervous system is active. When you eat, all of the components of your food are going to enter your bloodstream. Glucose, amino acids, fatty acids, all of that's going to spill into your bloodstream and start to rise. So we need to release insulin so that it can have its effect. Now, we don't always want insulin to be released. So how do we make sure that it's not released? How do we inhibit it? Well, when there's a drop in blood glucose, we don't want insulin to be released. But also, the hormone, growth hormone, it inhibits insulin. It doesn't so much inhibit its release, it does a little bit, but it inhibits its activity. And the sympathetic nervous system. When we have our sympathetic nervous system activated, it blocks insulin release. So what's the function of insulin? What does insulin do? First, glucose uptake, especially in muscle and fat. Let's think about how insulin works. What is its mode of action or its mechanism of action? Remember that from our last class. Well, remember that for cellular respiration to occur, we need two things. This goes back to Bio 137. For cellular respiration to occur, we need glucose and oxygen. So glucose has to be in the cell before we can use it to make ATP. How does glucose get into the cell? Well, oddly enough, even though our cells need glucose, glucose cannot enter our cells unless we give them the message to bring glucose in. How do we give them the message? Insulin. Insulin tells our cells to grab glucose from the bloodstream and pull it into the cell so that it can use it. So glucose uptake is a function of insulin. Next, glycogenesis that we just spoke about. The liver will pull glucose from the bloodstream and begin to string it together to make glycogen to store it. That way later, if we haven't eaten in a while and we need some energy, we have glucose on hand in the form of glycogen. Now, separately from that, at the brain, insulin plays a role in learning, in memory, in feeding behavior, and in neural development. So all of this important, especially in infants. So insulin, aside from its glucose function, it does have those other roles. So let's talk a little bit about some disorders that we can see with the hormone insulin. We're going to talk about almost all of the hormones, some of the disorders that can happen with them, and we will talk about two classes of disorders. Hypersecretion, what happens if there's too much of this released? Or hyposecretion, what happens if we don't release enough, or if we're releasing it but it's not having an effect. So insulin, if we release too much in hypersecretion of insulin, that will cause hypoglycemia, low blood sugar. Since insulin causes sugar in the blood to be pulled into the cells, well, if there's more insulin, we're pulling more glucose into the cells, causing low blood sugar. But the one that we're more familiar with is hyposecretion. What happens if we either do not produce enough insulin, or if we produce insulin but the cells do not respond to it? So hyposecretion of insulin causes diabetes mellitus. Diabetes mellitus. Now, most of us have heard of diabetes, but we are going to learn this semester, and especially in this chapter, that diabetes is not a disorder. It is a class of disorder. And there are lots of different types of diabetes. And I don't just mean type 1 and type 2 diabetes. We are going to learn about those in just a moment. But there are lots of other diabetes that have nothing at all to do with sugar or insulin. So first thing that I want to talk about is, why do we call all of these different things diabetes? Well, diabetes, like I said, doesn't mean anything about blood sugar or insulin. Diabetes means you're urinating a lot. Lots of different types of diabetes, all with different causes, but the one thing that they all have in common is you urinate a lot. So keep that filed away because we're going to refer to that throughout the semester. Let's begin by talking about the diabetes that most people are familiar with, and that is diabetes mellitus. This is the diabetes that you think about when you hear someone say, I have diabetes. And there are a lot of symptoms of diabetes mellitus. First, polyuria. Well, polyuria is increased urination. Poly means many or a lot. Uria, urination. Next, polydipsia. Polydipsia means increased thirst. Poly, again, lots. Dipsia, drinking. So you're very thirsty. Why are you very thirsty? Because you're urinating a lot. If you're urinating a lot, you're going to become dehydrated, so you get thirsty. Next, polyphagia. Phage means to eat. So polyphagia is increased appetite. You eat a lot. Decreased hunger. Well, this seems kind of counterintuitive. How can it be polyphagia and decreased hunger? Well, some diabetics with diabetes mellitus get very, very hungry. Some diabetics with diabetes mellitus lose their appetite almost entirely. So it could go either way. CNS depression. Now, that does not mean depression as in I'm sad. That means decreased activity of the central nervous system. So your nervous system starts to slow down. It could lead to coma if it slows down enough. Next, acetone breath and ketoacidosis. Ketoacidosis is what happens when we start to break down our muscle, our fat, various proteins, things like that, for energy. When that happens, because normally we're not supposed to do that, but when that happens, and it happens a lot, it actually produces things called ketones and acetones. So ketoacidosis, those ketones build up in the blood. They are low in pH, so it causes our body to become a little bit acidic. We enter a state called metabolic acidosis, but specifically that type of metabolic acidosis is ketoacidosis. And again, that happens when we start to break down fat and muscle for energy. And this is detectable on the breath. It starts to smell a little bit like acetone, fingernail polish remover. So diabetes mellitus, there's two categories of diabetes mellitus. There's type 1, which used to be called juvenile diabetes. It's not anymore. That's not really a proper term. But type 1 diabetes mellitus and type 2 diabetes mellitus. Type 1 is usually genetic. It's a type of autoimmune disease. Remember, autoimmune disease is when your immune system begins to attack your own body or a part of your own body because it thinks that it's foreign. In this case, it starts to attack your pancreatic islets, your islets of Langerhans. And here, if you don't have islets of Langerhans, well, you can't produce insulin. Or you're not producing enough insulin if there's still some islet cells left. But decreased or no insulin production. In type 2 diabetes mellitus, this is a lifestyle disorder. This is because of someone who eats poorly for a long time. Your cells become resistant to insulin. So even though insulin's being produced, your body isn't responding to it. That glucose is just going to stay in the bloodstream. Even though you've got plenty of it, your cells are not going to pull it in. This is reversible. Type 1, not so much. They're starting to get some promising leads, but for right now, type 1 diabetes, there's not really any way to reverse it. It's treatable with insulin injections and things like that, but it's not reversible. Type 2 diabetes mellitus is. If you're going to reverse type 2 diabetes, it's just a matter of changing your diet, changing your lifestyle, becoming more active, working out regularly, things like that, and it can reverse type 2 diabetes mellitus. All right, so now let's switch and talk about the alpha cells in the islets of Langerhans. Alpha cells are where the amino acid-based hormone glucagon comes from. Glucagon, you might have heard of it. Most people at this point have not heard of glucagon. It's essentially the opposite of insulin. So its stimulus is going to be a drop in blood sugar, decreased blood glucose. It's going to be activation of the sympathetic nervous system. But it is similar to insulin in that increased amino acids is also a stimulus. But as a general rule, when insulin is up, glucagon is down, or when insulin is down, glucagon is up. It is inhibited by increased blood glucose. It is inhibited by insulin. Remember, they are antagonists. And it is inhibited by growth hormone. Its function, we're going to have some crazy words here. You've hopefully heard them in Bio 137. First, glycogenolysis. Well, we talked about what glycogen is. And remember from Bio 137, lyse or lysis is breaking something down. So glycogenolysis is the opposite of glycogenesis. We're going to take that glycogen and break individual pieces of glucose off to release them into the bloodstream. And if we're releasing glucose into the bloodstream, what's going to happen? It's going to raise blood glucose. Next, gluconeogenesis. Gluconeogenesis. This literally means formation of new glucose. Neo is new, genesis, creation, glucose. That means we're going to turn something that is not a carbohydrate, it's not a sugar, we're going to turn it into a sugar. So break down fat, break down protein, but here turn it into glucose or something very, very close to glucose. That is gluconeogenesis. And third, glucose release from the liver. Well, in glycogenolysis, we're breaking that glycogen down into glucose, but we need to spill that glucose out into the bloodstream so that the body can use it. That's what part 3 there is talking about, glucose release from the liver. Now, hypersecretion, not so much an issue with glucagon, but hyposecretion can cause hypoglycemia. If we don't release enough glucagon, well, we don't have as much way to raise blood glucose when it gets low, so we can get hypoglycemia. Tie that together with insulin, hypersecretion of insulin, hyposecretion of glucagon, both can cause hypoglycemia. Now let's take a look at this. This should look very familiar. This is that negative feedback loop that we were talking about. Very similar to the shivering or the sweating from back in chapter 1. But here we're looking at glucose levels. And right in the middle, there's our seesaw, our teeter-totter, that shows normal blood glucose levels, about 90 milligrams per 100 milliliters of blood. What happens if it gets a little bit too high, or what happens if it gets a little bit too low? Because even though this is the normal, we're constantly teeter-tottering back and forth between the two. So stimulus over here, what happens if blood glucose gets a little bit too high, above about 90 or so? Well, the pancreas, specifically the beta cells, they're going to release insulin. And that insulin is going to, one, stimulate the cells to take glucose in. It's also going to stimulate the liver to make glycogen. When both of these things are happening, glucose levels in the blood are going to start to drop back to normal levels. So if it gets too high, insulin brings it back to normal. But at some point, we're going to overshoot and it gets a little bit too low. When it gets a little bit too low, the pancreas, specifically the alpha cells, are going to release glucagon, which stimulates glycogenolysis at the liver. Blood glucose starts to rise, bringing it back up to normal until we get just a little bit too high, and then we go through the top part again. So we're constantly going a little bit too high, bring it back down until we get a little bit too low, bring it back up. Constantly back and forth. That's our negative homeostatic feedback loop. But what about at the cellular level? Here we're seeing a cell. When there's blood glucose that's elevated, that glucose needs to get into the cells. So insulin allows that to happen. It actually inserts a protein there that brings glucose in. Glucose comes in, the cell either uses it for cellular respiration or to produce glycogen, if this is a liver cell. So here, now we've got glycogen stored for later. If we get low blood sugar, then glucagon is going to cause that cell to break the glycogen down. glycogenolysis. We're also going to convert fats and proteins into glucose, gluconeogenesis. Glucose starts to build up in the cell, and then it is released, bringing blood sugar back up. All right, so that's our talk about the pancreas. That's our talk about insulin and glucagon. So make sure to study those. Watch this a time or two if you need to. Ask questions as you need to. And I will talk to you next time.